Okay, Mr. Lim, the floor is yours.
Thank you. Okay, so let me just share my deck. Can everyone see my screen?
Yes, clearly. Yes.
Okay, everyone can hear me. Okay, good. Okay, so just a very quick introduction. My name is Ryan. I am a technical scientist for Thermo Fisher. So I support both the immunoassays as well as the immunotherapy portfolio. And I support the Asia Pacific and Japan region. So today, on behalf of our distributor, I'm here to talk more about two assays, basically our Procataplex, as well as our QuantiGene platform, which can be used on the LUMINEX platform. Okay, so that's what we'll be sharing to you today. So I shall first be sharing a little bit about the instrument, the LUMINEX instrument that you guys recently have, as well as the Procataplex assay and the QuantiGene assay. So as you can see here, there are two versions of the instrument, right? The first one is the MacPix, which has already been discontinued. It uses an image-based system. The system that you guys have in your facility and the university would actually be the flow-based system, right? So the LX200, along with the FlexMap 3D and the IntelliFlex system. So what happens is that basically this allows you to use, similar to a flow platform, to read the microspheres for you to detect multiple targets. So why is this instrument something that people want to purchase, or why is it useful, or what can you use it for? Well, at Thermo Fisher, we provide two major applications. The first application is, of course, to measure multiple proteins. You heard a previous talk that talks about the ELISA, sharing with you, how the ELISA works. And this assay works very, very similar to that. The second thing that you can detect, or the second application, is gene expression. You can measure mRNA or DNA as well with the same instrument. So what does this mean for you? It means that with the same kind of sample, you can get both the protein as well as your gene expression data. So this is just some images of data that we have gotten for you to demonstrate, from the same sample, so cell culture supernatant, from your cell culture, we basically take the cell culture supernatant and we measure the protein expression. And then we take the cells, we lyse them, and we ran the gene expression assay. So this is why we say, with a single instrument, you can measure both, you can quantify both protein and mRNA expression. Okay, so let's now talk more about the protein assays. So just want to share some of the highlights with you. Now, first of all, it is highly specific. It works just like a sandwich ELISA, as you heard the previous speaker mention. However, the unique thing about this is this allows you to measure up to 80 analytes in a single sample. Standard ELISAs allow you to measure a single target. So what happens when you need to look at multiple targets? You need to run multiple ELISAs. But you have different constraints like sample, the cost, time, and the accuracy of it. So this assay allows you to solve that. You need very low sample volume, about 25 microliters of serum or plasma, or 50 microliters of cell culture supernatant to run the assay for all your targets. So if you're running 10 targets, you just need 50 microliters of cell culture supernatant per well. Unlike if you're running 10 ELISAs. It reduces the cost, so the price per target is cheaper. So the more you put together, the cheaper it is. Ask yourself, how much does it take? How much does it cost to buy 50 different ELISAs? So we are a fraction of the cost. And lastly, it also saves a lot of time for you. You must think of it. How long does it take for you to run one ELISA? How long does it take for you to run 5 or 10 ELISAs, 20 ELISAs? So we can all do everything in 4 to 5 hours. So one day you can finish all the ELISA work you do. So how does this work? Okay, very simply put, the beads that we put inside for you are these micromagnetic spheres. So they are about 5.6 microns in size, and they are dyed in two colors. Okay, so there are two kinds of red and far red. And these are magnetic beads. And what Thermo Fisher does is we put our antibodies onto these beads. Because the ratio of the dyes are different, every bead is unique. It has a unique ID. So say, for example, bead number 12, maybe they put 80% red dye and 20% far red dye. So that would be bead number 12. Bead number 15 could be, say, 75% red dye and 25% far red dye. So the system knows what is the concentration, and it knows what's the ratio, and it will assign to it a unique ID. And on every bead, we will put in just the same type of antibodies. So for example, bead 12, as you can see here, is all IL-9. Bead 36 is all IL-6. So when you run the experiment and your laser starts to interrogate, every time it detects bead number 12, it will know, this is IL-9. Every time it reads bead number 36, it will go, this is IL-6. And it will make it such, so you can measure it accordingly. So this is the workflow. Very simply, you take the beads that we give you, you put in your analyte, and then you incubate about 2 hours, shaking, room temperature. After that, you wash it with our magnetic washer. Then you put your detection antibody, and then you incubate with shaking, 30 minutes. After that, you wash, you put my streptavidin PE, you incubate, same thing, 30 minutes. After that, you wash it out and you go and read. That's it. Okay? Very straightforward, very simple assay. And this is how the instrument reads it. The red laser, as I tell you, will detect which bead is it. And then the green laser will detect your fluorescence, the streptavidin PE. Okay? So it will detect that. And then it will give a signal, and the system will read the signal and assign it to the bead ID. So in a nutshell, what we're doing is this. When you look at the traditional ELISA, and say, for example, you're looking at 4 different targets, you need to run 4 different ELISAs. But for the Procadaplex, you can just put everything together in one well. So this saves your sample, it reduces your costs, it saves a lot of your time, but most critically, it becomes more accurate as well, because you don't have all your pipetting errors. The more you pipette, the greater the pipetting error. You have your interplate error, your intraplate error, your intersample, your intrasample. All these errors add together. But because you do all in one well, the comparison is better. So what do we have to offer for you? Okay, what do we have to offer? Okay, so we sell our kits in various formats. The first are singleplex. That means we sell individual targets for you, and you can put them in yourself. We sell you panels. So that means we already custom put all the targets for you, and we put them according to different application. For example, cytokines, growth factors, cancer, neural markers, so on and so forth. We have already put them together for you. If you're looking at SARS-CoV, we also have SARS-CoV panel, so on and so forth. And the last one is the more interesting one, is you pick and choose the targets you want. So we have a site that allows you to customize all your targets. So you pick and choose what you want, and you do not need to pay for targets that you do not want. Say, for example, we have a panel of 15 targets. Do you need all 15 targets? If you don't need to, say you only need 12 targets, you can tell Thermo Fisher to remove the 3 targets. Or say you see another target in another panel that you want. You can tell Thermo Fisher, I want that target, put it here. And we will do it for you. The cost is the same based on the number of targets. So it is not say this custom is more expensive than your panel. They are about the same price. So as I mentioned, all the panels are based on current literature. So based on what people are studying, we put the panels together. Like this is the immuno-oncology checkpoint panels that we have. I also mentioned to you, we can custom design. So all you have to do is come to our website, thermofisher.com/order/luminex, and you pick, the human, you pick your assay format, the instrument you have, and then you just pick your targets here. However, it will be easier if you reach out to NutriLab. If you don't want to do this, reach out to NutriLab and just tell them, hey, these are my targets. Help me out. They will have the abilities to also help you design the panel. That's how good they are. They can help you design it as well. So reach out to them, talk to them and ask them, this is what I want to look at. What do you have for me? Now, the main thing that a lot of people are concerned with, of course, is if I put everything together, is it going to be messy? Is there going to be cross-reactivity? The answer is no. We have already tested for cross-reactivity. We made sure that all our targets do not cross-react. They are unique. They will not bind to each other. So your signal is accurate. So we have experiments on this. Our data is also consistent through scalability. What does this mean? It means, for example, if you start with my bigger plex, say 30, 50 plex, you want to screen, and then you find only 10 targets are very interesting. You do not need to buy again 50 targets because only you want to see 10 targets. You can tell Thermo, I only want these 10 targets. Then we will just make it for you. And because we use the same antibody, your data is the same. As you can see in the center here, this one of them, the blue line, is multiplex. So that means we put all the panels, 34 together. But the red one, as you can see, is very similar. It's a single plex. So it's just one target only. So whether you buy a lot or you buy one target, the data is the same because we use the same antibody. And that is why our data is also mappable to ELISAs. When you move from the Procardaplex to the ELISA, say for example, you have a lot of samples and you just need to look at one target, you can ask for the same antibody as our Procardaplex so that your data is mappable across both the Procardaplex and the ELISA. Just want to share with you some papers that we have. So these are papers or publications that people use our kits. So this one is looking at immunoprofile in dengue patients. So the customer wanted to measure cytokine storms. So they started to look at all the cytokines that they want to look. And then they measure with different kinds of patient. Whether is it no dengue control, or they are people with mild symptoms of dengue, or they have very significant symptoms of dengue. And from that, they can see, which cytokines are more critical in predicting if a patient is going to suffer a very severe response. So these are just some of the things that you can do with the assay. People, of course, use it for vaccine studies. We have it for COVID. This one is for Zika. So when they study the Zika vaccine research, they give it to mouse. So we also have mouse targets. We give it to mouse and we measure also, what is the response after they get vaccines. Do they have inflammation or is it stopped because of the vaccine? So that is the protein assay. So I hope that that was a very clear explanation on that protein part. Just want to take some time to share with you, the gene parts as well, because we also have QuantiGene. Same instrument can do two things. So QuantiGene, we allow you to measure up to 80 genes in a well, okay? So why QuantiGene? Well, first of all, everyone likes protein because protein is functional. But what happens if we do not have the protein? There is no dengue protein ELISA in the market, right? Or maybe, they don't have specific proteins for certain diseases or certain proteins is not present. Then we look at gene. But why QuantiGene and not, say, qPCR? Now, first of all, QuantiGene is very fast. It has a very fast sample prep. So what happens? You take blood, you take tissue, you take cells, you lyse, and you run QuantiGene. That's it. You do not need to extract RNA. You don't need to purify, clean up. You don't need to count. You don't need to change it to cDNA, and you don't need to run qPCR. Okay, just lyse your samples and run. Or lyse your samples, keep in -80 until you get everything together, then you run the assay. So this is QuantiGene. You can measure up to 80 genes. So what does this mean? Again, same thing, right? You use lesser plates, better accuracy, because everything is in one well. Those of you who run qPCR, ask yourself, how many qPCR plates you run. It can detect very small changes because of how the assay is made, so up to 10%. you can also detect. Not like qPCR. qPCR must be fold change. This one, we do not need to. And it is very much like an ELISA workflow. So this is my workflow. Sample prep, just lyse your sample, and you can store it or run the assays. How do you run the assays? You put my LUMINEX beads and my mixture. So it has this capture extender, label extender, blocking, and lysate. You put them all together and you incubate for 16 hours or overnight incubation. Just shake in an incubator, 56 degrees. Next morning, you add the preamplifier. Just add, shake 1 hour. Wash, add amplifier, shake 1 hour. Wash, add label probe, shake 1 hour. And you run on my instrument. After that, you have all your data. So again, this is something that people use it for, like vaccine safety testing. So they wanted to see the expression levels, they can use it. So this one, as everyone knows, we all talk about SARS-CoV-2, right? So this is something that we made for a researcher using QuantiGene. So researcher wanted to see all the viral gene, so SARS-CoV-2 virus. So we have the open reading frames, right, and the main protease here. And then we want to measure the viral infection process. How is the virus infecting us? So we put all the viral infection genes. The user wanted to see the immune profile of the patient. So we gave her all the immune profile genes. Customer also wanted to see all the cytokine storm. So we give them the cytokine storm genes and then we put in the housekeeping genes. And with one assay, she can detect all of these at the same time. So that's what QuantiGene is. Okay. We also have the singleplex version, but that doesn't use your LUMINEX instrument. It uses a luminescence reader, so a luminometer. So that's what I just want to share with you again. QuantiGene is something that's direct from sample. You do not need to do any RNA purification. Okay, you can measure up to 80 genes in one well. You just need to tell us what genes you want, and we'll make them for you. Okay, so you must remember we own TaqMan. So TaqMan belongs to Thermo Fisher. So we use the same algorithm to design your genes. You can detect very subtle changes and you can work it in a 96 well plate. So how does QuantiGene work, right? We always talk about QuantiGene is so good. How does it work? So first of all, it uses something called branch DNA. Now, if this is familiar to you, it's because branch DNA has been in the market for many times. It's been in the market for a really long time. And it has been used also for a lot of IVD assays. So this is a technology or a platform that is well recognized. And that's what we do. So the thing is that qPCR uses something called replication. It replicates the number of transcripts. 1 become 2, 2 become 4, 4 become 8, 16, 32, 64, 1, 2, 8, 2, 5, 6, so on and so forth. But this replication is not efficient. It is not 100%. Sometimes it doesn't replicate completely. And that's why the errors come in. So that's why we always say qPCR, you can only trust if the fold change is at least 1 fold. But if you're looking for small changes, QuantiGene is for you. Because it uses branch DNA, it amplifies the signal, not the transcript. What do I mean? This is your target RNA. You cannot see it because it's only 1. So what QuantiGene does, it makes it brighter. So you just put in our first day 1 mix, and you just put them together, okay? And incubate overnight. Day 2, you add my preamplifier, my amplifier. You can see it's starting to become bigger, and my label probe. So it is like building a tree. You put the tree trunk, the branches, and the leaves, right? And you put your streptor vitamin P, and then you can detect the signal. You cannot see the seed on the floor, but you can see the tree that come out from the seed. That is exactly what branch DNA is. We make the structure bigger so the system can detect. Okay? So like I said, it is direct from sample. It takes any sample, blood, tissue, whole blood, RNA, cell culture supernatant, any kind of tissue, lyse it with my lysis buffer, and you can run it already. Just want to share with you, this is the protocol for whole blood. Finish. This is the entire protocol I have for whole blood. Warm my mixture, 37 degrees. Put my mixture with your whole blood. Shake. And then incubate for 60 degrees for 1 hour. 275 RPM. Finish. After that, you can run Quantigene or you can keep in minus 80 until you're ready to run. That is my lysis step. As I mentioned, we are TechMan. We own TechMan. So we use the same algorithms to design our assay. So you can see here what happens is that this is a gene. So we have our capture probes, which binds to it, the label extenders, and the blocking probes. So this is to make sure everything is locked in properly, so you don't have error. This gives you your accuracy because you pair up everything. Okay? Now, because it doesn't replicate, you have to surrender something. No technology is perfect. In this case, our limit of detection is higher, than qPCR. qPCR normally 1 to 2 transcripts you can detect already. For Quantigene, you need roughly about 1,000 transcripts. Seems like a lot, but if you think about it, if you're doing cell culture, okay, if every cell has 1 copy, 1 transcript of RNA, you only need how many cells? 1,000 cells. If your gene of interest have 2 transcripts in every cell, you only need 500 cells. So even though I say that the limit of detection is 1,000 transcripts, if you think about it, it's not really a lot we are asking for, is it? And in return, we give you really, really specific and accurate data. Now, what kind of applications do we have? I've already showed you we use it for vaccine. Just want to share with you some of the more interesting applications that people do. So the first one is your xenograph. So xenographing is difficult because you have human genes and mouse genes inside the same sample, and there's a lot of cross-reactivity. But because of the way Quantigene is made, with all the blocking peptides and label peptides, it's very accurate. You can tell the human gene and the mouse gene, and there will be no cross-reactivity. If you see the first column, this is a mouse gene, right? So background RNA, you see nothing. Human liver RNA don't have anything because it's a mouse gene. It cannot be seen in the human. In the mouse, you can see the mouse has it in the spleen and the liver, and in the xenograph, you can see the spleen and the liver as well. Now, if you see the second column, the human gene, again, background of nothing, but you see the human liver RNA, oh, we have a lot. But in the mouse, the spleen and the liver, we have very little. And then the xenograph, we have a lot again, telling us that the xenograph works. Not only that, you don't have the confusion with the mouse genes, and you can do it across. So this is, our xenograph model that people use it for. We also have a lot of others. We can verify genes for inflammation, DNA copy numbers, looking at tissue samples, FFPE samples, whole blood analysis. You can do toxicity studies. So the list is really endless. And we are constantly expanding what else people can do with our Quantigene platform. Okay. So just want to share with you though, we have multiple platforms. Everything is also Thermo Fisher. But Quantigene sits very nicely in the center area where you're looking between 3 to 80 genes. Okay, because then it will save you a lot of time in running on a plate. So again, reach out to your reps, and they can definitely help you with this as well. So just want to have my takeaway message, right? So first of all, I just want to share with you, nowadays people are looking into cytokine expression, especially in the field of immuno-oncology, inflammation response, like what COVID is having, where you have your cytokine storms. So the thing is that cytokines don't work alone. It is many cytokines. And when you start to look at cytokines, right, people will ask you, when you publish a paper, they will say, why do you only study this one cytokine? You cannot. Everything works in tandem. So there's always multiple function, multiple cytokines. And this is a very key area, especially now in the times that we are in, with all this infection, with cancer on the rise. This is something that people like to look at. Second, why this platform is because you need to be more consistent. You need more efficient platform. so that you can get your solution faster. Everything can be done in one day, and you have greater accuracy, right? It's very, very accurate. It gives you the ability to see things properly. You don't have all your pipetting errors. And lastly, is you also have to see, which platform is suitable for you. As I mentioned earlier, Quantigene has looked into 3 to 80 genes. We have other solutions as well. Same as the ELISA and the Procadaplex. If you're looking at a single target, don't get the Procadaplex. The Procadaplex is for multiple targets. If you're trying to understand the pathway, then yes, get the Procadaplex. So always see, which platform is better for you. So talk to your sales reps, talk to our distributors, and they can definitely help you with that. Okay? So with that, that's the end of my session. Thank you very much for your time. I pass it back to the moderator. Thank you.
Thank you, Ryan. Nice presentations. Baik, Bapak Ibu semua, saya akan memulai membuka sesi pertanyaan. Akan saya buka untuk 3 pertanyaan